My name is Dr. Stephen Pelech. I'm a professor in the Department of Medicine at the University of British Columbia, where I've been on faculty for about 36 years. I'm also a co-founder of two biotech companies, Kinetek Pharmaceuticals and Kinexus Bioinformatics, and I've been the president and the chief scientific officer of Kinexus for over 25 years. And our company actually undertook a clinical study to look at the levels of antibodies against the SARS-CoV-2 virus in about 4,500 people. And so we started this study actually in March of 2020, and we did it over a 3-year period. So I've also been involved in the development of testing drugs that can inhibit the replication of the SARS-CoV-2 virus. In addition to that, I'm one of the founders and the vice president of the Canadian Citizens Care Alliance. We were formerly called the Canadian COVID Care Alliance. And I'm a co-chair of the Scientific and Medical Advisory Committee of what we call the CCCA. And that committee has been meeting for well over 3 years, almost weekly, with about 30 other scientists and professors and healthcare professionals from across Canada. And we've reviewed the scientific literature with respect to published accounts for research related to COVID-19 and the vaccines. And in addition, we review public health documents and information that comes from the companies directly. And we critically analyze it. And we have a lot of debate and discussion within our own group. And we've actually written articles and recently 2 books actually on COVID-19 that are now actually available. So we've had a lot of peer review within our own organization. And our objective is to make sure that the information that goes out there is scientifically sound and unbiased. The CCCA itself does not receive any funding from government nor from any companies. And so we're independent, and most of us that are doing this, well, all of us on our committees are volunteers. We don't actually get paid for this. And that's sort of our way of trying to help in dealing with first the COVID-19 crisis, more of a crisis from the public health response than from the virus itself. But also we recognize that there's a lot of changes in our legislation that's happened or being proposed that will dramatically have an effect with future infections. So we're seeing an erosion of what we think are civil rights, actually, from these new legislations.
So we've chit-chatted, so I'll just dive right into it and you can just chat away.
Yeah.
Okay.
I'll try to answer your questions as best I can.
Yeah. Can you explain what the spike protein is in the virus?
Yeah, the spike protein of the SARS-CoV-2 virus is like many other coronaviruses. It extends out from the surface of the virus, and it allows it to interact with proteins that are on the surface of what we call the host cells, the cells that they're going to infect in the body. And in the case of the SARS-CoV-2 virus, it primarily is targeting the ACE2 protein, which is involved in control of blood pressure. So when it binds to it, it actually interferes with its function, which is normally to degrade a hormone called angiotensin 2, which actually increases blood pressure.
Okay. So what did they do then with the vaccine to mimic the spike protein, or can you talk about?
Well, the genetic vaccines, there was really 4 of them that were introduced. 2 of them are what we call mRNA vaccines, and the other 2 are adenovirus vaccines. In both types of situations, it's the genetic material that allows the cell to use the gene, basically, that encodes the protein, the spike protein, so that it's actually produced inside of the infected cells. So we're not actually putting the spike protein in with these vaccines. We're actually putting the material, the genetic material that instructs the cells to manufacture that protein. So in the case of the adenoviruses, they're using DNA, which is then transcribed into what we call mRNA, messenger RNA. DNA is actually very stable, but the messenger RNA is less stable. However, these vaccines, in the case of the mRNA vaccines, are engineered with a form of the mRNA with a substitution for one of the nucleotide bases called uridine. So they do 1-methyl pseudouridine. And this actually makes the mRNA much more stable. So this mRNA would come in with lipid nanoparticles to penetrate through the cell. And then inside the cell, there's protein synthesis factories that take that mRNA and make the spike protein. And that spike protein gets imported to the surface of the essentially infected cell with the lipid nanoparticle. And then on the surface of those cells, what happens is the immune system will have other immune cells, macrophages, neutrophils, dendritic cells. These cells seek out foreign invaders, proteins usually on the surface of these viruses or bacteria or fungi. And in the case of a cell that has been infected, these foreign proteins will be expressed on the surface of those cells. And so in the case of the vaccine, we're kind of mimicking an infected cell. So the immune cells attack it. They cause damage to the cell. They create debris. We call these exosomes. And these tiny little fragments of the cells that are coated with these viral proteins, in the case of the vaccine, would be the spike protein, are then gobbled up. They're digested inside those immune cells. And then they're presented on the surface of the immune cells with other immune cell proteins called the major histocompatibility antigens. And then what happens is they are these antigen presenting cells move to like the lymph nodes where they will encounter T cells and B cells of the immune system. And then those, if it turns out that some of those cells have an affinity for that fragment of the virus, while presented with these other special proteins from the immune cells, it stimulates them to grow and divide and create a whole clonal army, essentially, of identical B cells and T cells that recognize that particular foreign antigen. So what happens is the T cells attack cells in the future that have been infected with that infectious agent, in this case, spike protein expressed on the surface of T cells from a virus infection, or the vaccine in the future sets them up for actually being attacked. And then the other is the B cells. These cells produce antibodies, and antibodies will be very specific for that viral fragment. And then what ends up happening is the antibodies will stick both to the virus as well as to cells that are infected with the virus. And so when you have antibody-coated cells, through the interaction with this foreign protein, you set up what's called the complement system in your blood, which basically blasts holes in cells to kill them because they think they're infected with a virus. Or they also trigger the attraction of other immune cells that would again attack those cells and gobble them up. So this is, in principle, it's pretty straightforward for someone with immunology knowledge to recognize that you actually are setting up your own body cells for attack by your immune system when you're producing a foreign protein on the surface of your own cells, which is exactly what these vaccines do. And the problem is that when you inject into your deltoid muscles, the lipid nanoparticles, about 76% of them apparently are actually gone from the site of injection, and they travel throughout your body. And what most people don't understand is that when you get a shot of, let's say, a Moderna vaccine, within that shot, you have tens of trillions of lipid nanoparticles, each about the size of a virus. each with about 5 to 10 copies of the gene that actually encodes the information to make the spike protein. And so it's traveling throughout your body. It goes through the blood-brain barrier. It can go down the umbilical cord into a baby. So you're really seeding these, not just in your arm, but everywhere. And the places where they tend to concentrate is the liver and the spleen, the adrenals, and also the ovaries is another place where it gets concentrated. So that, of course, creates its own concerns, but you're setting up for an immune, autoimmune disease in a sense. Now, how the immune system responds to a very large, what appears to be an infection on a repeated basis is that it begins to try to sort out, well, what's a foreign threat and what is something that's naturally in the environment, like in the food that you eat, like pollen that is seasonal, even the proteins on the surface of your own body cells. So the consequence is that with repeated boosting, you actually get an immune shift. The type of antibodies that you produce in response to an injected vaccine that's going to get into your circulation is what we call an IgG response. And initially, you have an IG1 and an IG3 response. So these responses are pro-inflammatory. That means that you will actually have your immune system attack. It'll mount a defense against the infection. However, with repeated exposures, you tolerize your body to the foreign agent. So it could be a switch now, and we see this clearly in the COVID-19 vaccines with the booster shot, the third time you've been vaccinated, that we get a switch to the IG2 and the IG4 class antibodies. And these antibodies are anti-inflammatory. They actually downregulate the ability of your immune system to recognize the foreign threat. So this explains why, for example, in the Cleveland Clinic study that was done with about 51,000 healthcare workers in the healthcare system, about 200 hospitals, that they followed them starting in September of 2022 for 6 months. And what they found was those that were the unvaccinated healthcare workers had the least chance of actually getting COVID again during the next 6 months. Now, again, this would be during flu season. And with every extra shot that those healthcare workers experienced, their risk of getting COVID actually increased. And there's been many studies since then that actually confirms the same. Most of our vaccines don't work this way. If you get a typical vaccine in the past, which is much safer, what you'll see is that maybe 50 to a few thousand copies of a weakened strain of the living virus would be injected into a person. And because it's so weakened, it actually allows an immune response, without making you sick, but it can be sustainable for even decades. So, we see this like polio and other vaccines. And so when you compare that, and by the way, each virus will only have like one copy of its genes to make these foreign proteins. Again, when you're comparing that to tens of trillions of lipid nanoparticles with a single injection, it's a completely different ball game. The immune system has never seen this before naturally from an infection. If you had that level of what we call like a viral load, you'd be so sick. And so what ends up happening is with many of these vaccines that are the COVID-19 genetic vaccines, from Pfizer or Moderna, what you find is that about 70% of the people that are actually injected do feel it. They have an adverse effect. In most cases, it's fairly mild. It's sore. But what we find from the clinical studies is that about 5% of the people that were in the original phase 3 clinical trials with the Pfizer vaccine, they actually had adverse effects that were severe enough that they required to see a doctor. So we do not see this with regular vaccines. And then the other thing that kind of illustrates the problem that you have these adverse events is looking at the US VAERS system. This is the Vaccine Adverse Effects Reporting System. And in that system, we have more than 36 thousand deaths that are recorded. But let's say in the United States alone, there's somewhere around 19,000 deaths reported associated with the vaccines. Most of these are within, a few weeks of the vaccination. And what you have to do is to kind of put it into context. And so with over 1 million reports of vaccine injury in the VAERS system, which is thought to actually dramatically underreport the total number of cases. Some people argue that it's only about 2% of actual cases get reported. So you could take those numbers I gave you and probably multiply it by, at least 40-fold. Well, what happens is if you take all of the vaccines that have been reported in that system since over 32 years ago, and you combine it for about 80 other vaccines put together for all the adverse reactions reported in deaths, it is still less than the total number of adverse reactions and deaths reported with just the 3 vaccines that were approved in the US that were genetic COVID-19 vaccines. They did not approve in the United States, for example, the AstraZeneca vaccine. Yeah, we did in Canada, but there was about 14 countries in Europe, for example, that also did not approve it.
Why did we in Canada?
Well, I think we were very desperate early on to take on this threat, and we thought that vaccination was really the main solution to the problem. Actually, as I've outlined, it probably exacerbated the problem, but we took it on. In the summer of 2020, the Canadian government had negotiations with about 7 different vaccine manufacturers. So they didn't actually have a product available at the time, but nonetheless, we got into commitments enough that would have allowed vaccination of every Canadian citizen about 4 times. And then the approvals of those vaccines didn't actually happen until the beginning of December. So we have our government negotiating contracts to purchase these vaccines, and then having our own regulatory agency, Health Canada, go through a review of what would have been only 2 months of testing data in people, then making the decision under interim order in Canada. In the United States, that the emergency use authorization. And what happens is in the interim order in Canada, you actually don't have to demonstrate that the product is safe, nor that it's effective. What they do is, in the way that legislation is written, you have to ascertain that the threat of, in this case, the viral infection and the consequences is greater than what you think the consequences would be of a vaccine. And we think of vaccines in the traditional sense, like I was explaining earlier, that, attenuated viruses. And then they compared that to early data where they were contemplating that the death rate of this SARS-CoV-2 virus, population-wide was, 3% or higher. And of course, as it turns out, that's completely untrue. If you're someone, for example, that's under 65, your chances are probably less than 5 in a million that you would die from it. And most of those that would die from it have comorbidities, on average about 4. So the risk would be higher for people that are much older, but then the risk of death in general is higher for people that are much older. And so if you look at the average age in Canada of someone who died from COVID-19 or with COVID-19, it's about 84, whereas the average life expectancy in Canada is about 82. So there's nothing really actually unusual here. they'd be susceptible to probably any infection. And what we saw in 2020 and 2021 was a dramatic decline in the number of cases of influenza. And so when we track, influenza in Canada, we track influenza and we also track pneumonia deaths. And as it turns out with COVID-19, most of the deaths were actually due to pneumonia that happened as a secondary infection, not so much from the virus itself. And in fact, when you look at the data for people that went to hospital in Canada, and we certainly have this data for B.C. Bonnie Henry, our chief public health officer, disclosed it, that most people that went to hospital actually did not have COVID symptoms. They were there for something else. And then, of course, they got tested, and either they acquired the infection in the hospital or they had the infection and they were asymptomatic. So, in fact, we know that most people, that were certainly children, were completely asymptomatic when they were infected. And probably about 40% of adults that were infected had no symptoms of COVID-19. And when you don't have that many symptoms, you're not particularly, contagious. There is a risk that you could still spread it, but it's much, much lower than people who are actively sick and producing large viral loads that can be aerosolized and then breathed in by other people. So we're seeing a erosion of what we think are civil rights, actually, from these new legislations. For example, Bill C-293, the Pandemic Preparedness Bill, which actually has no plan of what they're actually going to do. But what it does do is it empowers government to have even more powers to take out the rights of Canadians. And so that's problematic. And it doesn't really encourage more research in the area. And it doesn't come up with alternative things that we should be doing to improve the health of Canadians so that we're actually more resistant to infections in the future. So yeah, it's got a lot of shortcomings.
Were there two highlights from you that stuck out, as in, what is this even doing in here in that bill?
Well, the thing about the bill that's probably the most problematic is that what we're doing is we're allowing the World Health Organization, which is, not an elected body, which has a lot of funding from China, but also from private, enterprise like the Bill and Melinda Gates Foundation. And so they've promoted these vaccines. And when you have a situation where the WHO would then declare a pandemic, and they can do it with very little cases actually worldwide, to declare a pandemic, it would immediately permit national governments to then implement basically these mandates on the fly, and so there's no scrutiny. Usually it'll be done with advice from civil servants that are non-elected, and it takes the in the case of healthcare delivery, the power away from the national government, the federal government, to actually what was normally should have been in the provinces. This now gets really under the control of the federal government, which itself is going to be influenced by these international organizations, which again have different agendas. And you can't necessarily apply these same strategies in different places in the world, but yet the objective here is to make it very broad. Like, now, let me give you an example. When you look at how we responded in Canada to the SARS-CoV-2 infection, COVID-19, we did all these lockdowns, and we closed businesses, and we restricted travel, and we did a lot of things that we thought would mitigate basically the spread of the virus and its effects. However, another country, Sweden, took a totally different strategy. They did not do the lockdowns. They did, certain things to try to protect those that were their most vulnerable, and they encouraged their citizens on their own to take measures that would be hopefully protective, but they didn't mandate it. And so children still went to school. The shops were still open. People could still travel. And in the end, when you look at the total number of deaths that occurred in Sweden and the impacts on, their healthcare system, they had a better outcome than the United Kingdom and France and Germany. And so when you look at many of these other European countries, they actually did better in the long run than the rest of them. And they did, basically allowed natural immunity to occur, but what we would consider herd immunity. And when a large percent of the population has been infected, you actually prevent less likely that that virus will spread, but also that it'll mutate. Because when you have a pressure with a vaccine, much like an antibiotic with bacteria, if you constantly have this antibiotic in the environment, you select for viruses that are resistant to that antibiotic. And in a sense, that's what we've been doing with these vaccines. We've been selectively allowing those mutations of the SARS-CoV-2 virus, these variants, especially variants of concern, to mutate and evade the previous vaccines that we've had. So these vaccines, the COVID-19 vaccines, are just not that effective that were originally developed for the Wuhan strain, the first strain that came about. So this is very interesting because if you look at SARS-CoV-1, this is a related virus to SARS-CoV-2 that was responsible for COVID basically 22 years ago. In Canada, we had about 147 deaths, a little over 700 worldwide from that. Again, it came from China. We know it was from a bat virus because that's the closest similarity. Like if you look at SARS-CoV-2 virus, what we find is that genetically, we have bat viruses that are 96.8% identical in their structure to SARS-CoV-2. So we know that that's where it came from, but the evidence is really pointing that that virus was further engineered to take portions of SARS-CoV-1, which made it able to infect humans, introduced into this bat virus, and gave rise to SARS-CoV-2. But then as SARS-CoV-2 infected our population, it underwent mutation. So now we have the latest variants, within the Omicron family. They're still about 96% identical to the original Wuhan strain, but they're more infectious, but they're also more benign. They're weakened viruses. And so it's actually now a natural kind of infection that, people get. They don't get as sick, but then they get that strong immune protection against future infections. So what's interesting with SARS-CoV-1 from 22 years ago is that we didn't do anything. We hospitalized people that were infected, and there were some deaths, as pointed out, but it disappeared in about a year off the entire planet, apparently, without any vaccines, without any special medications that targeted that virus specifically. So what happened? Well, I think it's very much like what we saw happen with SARS-CoV-2. We had this the mutation of the virus over a period of time from the Wuhan and the Alpha, the Beta, and then eventually, Delta strain. And the Delta strain was more infectious than the Wuhan strain. And then what happens is these variants displace the earlier variants because more people are likely to get infected with the more infectious version that doesn't make them sick. And then what happens is they'll go out and spread it because they're not aware that they are carrying it. But when Omicron came along, this was even more infectious and even more benign. And so in November of 2021, the dominant variant was the Delta variant in Canada. And then in the space of a month, in December of 2021, all of a sudden it was Omicron that was everywhere. So we can see that within a population, you can have a huge shift and the emergence of new variants that are actually less likely to make you sick and more likely to be infectious. And they become endemic. And we have at least 4 cold coronaviruses that are endemic in our population that's responsible for about 30% of common colds. They make you ill for a couple of days. And that illness is actually a reflection of your own body's reaction to actually boost your immune system and to clear the infections. So you have postnasal drip and you're coughing and your temperature is higher because your immune system works better with a higher temperature. So these responses are the same responses you have to other types of respiratory infections. And so what's happening is you get it, but it's not deadly. You're very unlikely to die from a common cold. And this is sort of where the SARS-CoV-2 virus is now has evolved. You know, it's been selected for, again, because of this vaccine pressure, to be the dominant ones now. And in fact, what's happened is because this virus undergoes mutation all the time, we actually have a whole bunch of different variants right now that none of them are particularly the dominant one because they all are very similar. They've kind of maximized out how infectious they can be and how benign they can be. So whereas we had discrete variants early on in the pandemic that predominated, we don't have that now. Again, because it's really endemic in the environment. And it's also not just in humans. I mean, there's so many animals that have been, even early on in 2020, animals that had SARS-CoV-2 virus. So even if you could wipe it out from the human population, it would very quickly come back in from these animals that are, you know, we call them animal vectors that would be carrying it, and you'd have zoonosis going from an animal to a human. So that's sort of where that's got at this time.
I was living in Hong Kong during the first one, and yeah, it was we took care. We wore masks if we went out. You didn't know, but yeah, it was we still went to work. Kids still went to school. We did everything. I thought they said it was civet cats in the market that
They thought that was that, and then it's also thought to be bats, and then
yeah.
it's pangolins, and you know, it's in fact, this is the interesting thing. There is originally they looked at over 50,000 samples collected in that Wuhan market area, and they couldn't find any evidence in bats for the SARS-CoV-2. They're not supposed to be carrying bats anyways in that market, but nonetheless, with these live animals, they went back in very, very hard. They have data from samples collected in January of 2020, and they can find some cases where there seems to be SARS-CoV-2 there. But the problem is you don't know if that's from people that then introduce it to the animals or whether it was in these animals. And there's very, very few cases. But what we do know is that in October of 2019 in Wuhan, they had the World Military Games. They have like about 100, over 100 countries, I think around 110 countries, actually send their soldiers there to do the equivalent of the Olympics. And they have these games. And at the time, the people in Wuhan were actually, there was a sickness going around. It was, some people say it was almost like a ghost town. We know from U.S. military surveillance that there was a large number of people that were going to the hospitals. They looked at the parking lots from satellite technology, and they went on the internet, looked to see how many times people are are Googling things like, flu or sickness so they could tell that there was a huge traffic. There was definitely something going on. And then what happened was when the Canadian soldiers came back to Canada on the planes, a third of them were really, really sick. They were at the back of the plane. And then when you start to look at the data in other countries where they start to actually look at blood samples using PCR tests, they can find reports now in samples that go back in October and November and December, indicating that in fact, probably through the military games, these people coming back, they were spreading it actually in their communities. And so even in, we think of Italy being one of the first places in Europe to be infected, but they have data now that show that the viruses are even 3 months beforehand. And so, the thing about Italy is that when you have manufacturing there, what often happens is you have Chinese factories in Italy, and you have about, tens of thousands of workers that are actually from Wuhan that go back and forth to the factories in Italy. So if you have a virus, in Wuhan, chances are very high that it's going to eventually get into place like these factories in Italy. They do this because, they can say that the garment is made in Italy, right? So what happens is it's not hard to understand how it can spread so quickly. One of the things that's really I find fascinating is officially in Canada, we're saying that this pandemic started in mid-March is really when it took off. But in fact, when we tested people, and we did this with the B.C. Women's and Children's Hospital, they had about 276 healthy adults, about half of them were healthcare workers, and we tested with our test at Kinexus, plus a different test that was done on the same samples. So the data confirmed each other. 90% of the people that we tested had antibodies against multiple proteins from the SARS-CoV-2 virus, indicating that either they had been infected with a virus very similar to SARS-CoV-2, or they were actually infected with the virus and didn't know it. And so when you look at the BC Center for Disease Control sequencing of the virus, by mid-March, that virus was primarily the variant that was coming from Europe, across Eastern, North America, and then gets to Vancouver. Well, I've just explained to you how you can have within a matter of months the displacement of one variant with another variant. So I think what happened was we had actually previous exposures. We didn't know it. Most of our population developed antibodies, and these were protective. And, it's kind of fascinating. I mean, if you think of a virus that's in China, the most likely place it's going to show up first in North America is Vancouver, because we're the gateway, to the Orient. We have, where I live in Richmond, BC, our population is 54% plus Chinese. And, these are very successful people. They travel back and forth to their businesses. And the incidence of COVID-19, when they started being able to track it, using the PCR test, which tells you whether you have active virus present or not. It doesn't work after you've recovered. They had the lowest rates of COVID-19 in British Columbia. So, it doesn't make sense, but it does make sense if they were infected early, they were fairly resistant, and as a consequence, we didn't really have that many cases. I mean, when you look in BC, early in the pandemic, in the first year, where we had our first wave officially, and of course, that wave is really a reflection of our introduction of the testing, right? So you may have had most of the cases, but you didn't know it Then as your testing goes up, you get the beginning of a wave. Then as that wave subsides, then you see it drop down. Well, with that kind of a wave, most people were probably infected long before that, and they just didn't know it. And we had at the peak of that wave about 400 people that would be actually in hospital in in um, in British Columbia at a time for COVID-19. We had 5,000 beds in the province. We emptied half of them to make way for what we thought was going to be this onslaught of COVID cases, but only about 400 at a time at the peak. That's what it was. So it actually hit us, it was pretty mild. So I'm putting into perspective the fact that we had this concern about people dying. This was going to be like, the 1918 influenza pandemic. And we took all of these measures, but in fact, the measures that we took caused more problems. The lockdowns, these children that couldn't go to school, even the masks turn out to be more problematic than protective. There's been many studies, a Cochrane study, meta-analysis of combined results from many studies. They're published in peer-reviewed journals, basically show that there's no benefit, even with N95 masks, in reducing the transmission of the COVID-19 vaccine or you being protected from it. And in fact, if you understand the physics of how these masks work and the pore sizes, the average pore size is about 30 microns. Microns about a millionth of a meter. Well, what you find is that these viruses are only 0.15 microns, and they might be in an aerosol, but these aerosols, if they're greater than 60 microns, they fall right to the ground. After that, they can remain suspended in the air with the virus on it, but they evaporate. They get smaller and smaller as time goes on. So they can go right through the masks, like mosquitoes going through a chain link fence is often one of the analogies. So there is actually no real protection from these masks. However, there are problems with these masks because when you wear them, especially if they're tight fitting, you're rebreathing your own air. So the concentration of oxygen in the atmosphere is about 21%, and the concentration of CO2 is 0.04%. And when you breathe out, you're actually breathing out about 14 to 16% oxygen and 5% CO2. And so when you are breathing this out in an obstruction, you're rebreathing some of that air. So over a prolonged period of time, you're actually lowering the amount of oxygen in your tissues, your blood, and then your tissues. And you're increasing the levels of CO2 that are actually in your tissues and blood. And of course, your brain is very active and it uses a lot of that oxygen and a lot of the energy that you produce from your food. 25% of it, 20 to 25% is used by your brain. So you get brain fog. It's not getting enough energy. And so your learning, for example, if you're in school, is going to be compromised. Plus, wearing a mask is a very powerful visual signal that, there is a deadly agent in the environment that you need to be scared of. And that actually has profound effects on psychologically, especially children. And we know that when you are stressed, your immune system is depressed. So you're actually more likely to be sick, including cancer, because your immune system is also protecting you against cancer. And so when you're screwing around with the immune system by, doing these vaccines, and you're also creating very difficult conditions, especially that are going to be the most harsh on those that are marginalized economically, then you create a situation where they're more prone to actually developing illness. So we may have probably had more illness as a consequence, and deaths from the imposition of how we handled the COVID-19 pandemic, including the vaccines, than from the actual virus itself.
I just want to go back to the mRNA technology.
Right.
So obviously it was being developed and worked on before any of this happened, I guess. Maybe could you talk
Yeah,
about its inception? What were they going to use it for? Why all of a sudden did they come in and use this technology? Like, what about a traditional vax? Was it all just impatience and panic and
that's right. Well, first of all, what's been proposed to people with respect to the RNA vaccines is that the technology has been in development like for 40 years, including like the lipid nanotechnology. And that's true that the lipid nanoparticles were developed in Canada, largely through the work early on that stemmed from basically at the University of B.C. And what happened is Moderna and and also Pfizer both licensed the ability to use the lipid nanoparticles from UBC and from companies that were spun out from the investigator that had worked on that. And basically, the RNA itself, we've known about RNA for pretty close to 70, 80 years. The main application of the RNA in terms of being a therapeutic was more for cancer applications, and it's using what we call antisense RNA. So antisense is where RNA has a structure with nucleotides, and they have a sequence that's going to dictate of those nucleotides the sequence of amino acids that gene encodes. Now, you can have an identical structure that's the opposite nucleotides that will bind to the RNA, so the RNA cannot be used to make protein. So we call this antisense RNA. So there's been over 20 years of research looking at antisense RNA as a therapeutic, which is injected basically to hopefully get into cancer cells to block cancer proteins. So that technology has been successful in limited applications. And after 20 years of companies working on this, we have maybe a dozen of these drugs on the market now, but they work completely differently. They're to block the production of a protein. They're not there to create the protein. So the technology has been used in one instance for a vaccine, and that was about a year before COVID-19 that it was approved, and it was really for the Ebola virus. It's that vaccine is not available in North America, but it was in Europe approved. And the problem is that in the animal studies, when they infected them with Ebola virus, half of them, even though they were vaccinated, died. So as a consequence, there was no human studies with the approach of using the mRNA to then infect, basically vaccinate people. Now, the vaccine has been used, but there's no data available to establish its effectiveness. But what they have done is they've given it to people, and they didn't see, or they didn't think they saw, adverse effects. But people are not usually being infected with Ebola. The thing is, the more deadly the virus is, the less likely it actually spreads. So we only have one predecessor that's actually a vaccine that's not really tested for its efficacy, but the data, we don't know about its safety because it's a relatively small number of people that were actually tested. But it is on the market, and people will get it under circumstances where they think they're at risk. So there was no precedent for this.
Okay,
And
sorry. So if you need it for this, you're talking about the vaccine for Ebola,
for
right?
Ebola,
So
that's
is
correct.
that because talking about how more of these boosters
Mhm.
can increase the chances of adverse
Right.
effects. So
You could
my
have
question
one shot.
is this Ebola a one shot, one and done, and that's it?
That's
Different than
pretty
what we
much it. Yeah.
okay. Can you talk
So,
to
so at most two shots, but never three. So,
sorry, can
and
you just say
again,
that clear? So can you just say that clear so I could use a soundbite of that sort of you could say
sure.
the
Well,
difference between that Ebola. Yeah.
yeah. So this one virus that so vaccine that has been approved has for Ebola has never been tested for efficacy, and at the same time, we don't know how long it lasts, even looking at antibody levels in response to that vaccine. And we don't have any long-term safety data because it was really only introduced into the population about barely a year before the SARS-CoV-2 vaccines were introduced. So very limited data. For the SARS-CoV-2 vaccines themselves, as I mentioned earlier, they're actually uh, only had 2 months of testing data at the time they got the interim order or the emergency use authorization in the US approvals. The study went on for another 6 months for the Pfizer vaccine, for example. And based on that data, they later approved it after the interim order approval had expired. And so that clinical study, which was done with about a little over 40,000 people, 20,000 of them vaccinated, 20,000 of them were not vaccinated. They actually mixed the information from, during the study, they started having uh, teenagers, data of teenagers mixed in, and they're the least likely to actually get, sick and uh, and have adverse effects. So what happened was 2 months into the study, they unblinded the study. So everybody knew, including the doctors and the patients or participants, whether or not they actually had the vaccine or they didn't have the vaccine. And what they did was they allowed people to actually, that weren't vaccinated, to then get vaccinated. So 80% of the people in that study went on and got vaccinated. And the problem is we can't tell now what the long-term effects are of these vaccines in that population because we don't have much of a control group that's left. And what's interesting is when you look at the vaccine injury in the clinical study, firstly, more people died in the vaccinated group than the unvaccinated group. Not by much. Actually, very few people actually got even sick during the 6 months at a time that we were supposed to be having, well into probably the second and third wave of the COVID-19 pandemic. And so, but this is what other people are not aware. Now, we have relative efficacy and we have absolute efficacy. And relative efficacy basically is looking at, well, of the people who got sick, how many of them were vaccinated and how many were not. And based on the initial data, the relative efficacy was probably close to about 90%. And with subsequent vaccine shots, it actually seems to been getting lower and lower, and the duration of that protection gets shorter and shorter. But with absolute efficacy, which is really the question that we want to answer, and which is normally the information that would be provided with a drug or even a vaccine, is well, if you as an individual over 6 months in that period of time, what was the chances that you would actually get COVID-19 if you were vaccinated compared to if you were unvaccinated? And as it turns out, um, less than 1% of the people that were in the study actually got COVID. So, well, a few percent. And what happened was when you compared the people that got COVID to the people that were vaccinated, and were unvaccinated, the difference between the two groups, what we call the absolute risk reduction, was 0.8%. So what that means is if you were to vaccinate everybody during that 6-month period of time, you would reduce the incidence of COVID-19 in the population by 0.8%, not 90%, 0.8%. So that was one, important, consideration. But nevertheless, the thought was, well, if we vaccinate anyone, we can have herd immunity. And at first, we were hearing that, well, if we can get at least 60% or 70% of the population, this will do it. But of course, by the time you have about 88% of the population vaccinated, especially in places like British Columbia, which had the highest vaccination rates, the incidence of COVID-19 actually increased. So this is exactly opposite to what you expect for vaccines that work. And so then the explanation was given, well, yes, we recognize that it doesn't prevent the transmission. You can still get sick if you're vaccinated, but you won't get as sick. But see, the problem is, at the same time, you've got a virus that's evolving, become more benign and more infectious, and you've got a population that's been exposed to those earlier variants to which they developed antibodies that have natural immunity. So there's no way of telling that these vaccines actually reduced your chances of getting sick and ill. And then when you look later in the pandemic, in some cases in Canada, over 95% of the people that are in a hospital were actually more than double vaccinated. So that doesn't tell you that it actually reduces your severity. So yeah, it's amazing, basically, the spin that's been made to try to really promote these vaccines. And we're still doing it today from public health. This is what's amazing, despite all of the data showing how ineffective it is, how we're seeing increased negative efficacy with more booster shots, and how we have many people with severe illness for a vaccine, especially for healthy young men, for example, 12 to 29, they have about a 1 in 2,000 to 1 in 6,000, depending on the study that you look at, of developing myocarditis from the vaccine, where, again, their chances of having an illness that's going to be persistent from an infection with the virus is probably, in the order of 1 in 100,000. It's just crazy. But that's where we're at now. And then, of course, you have blood disorders, and we have increases in all-cause mortality, not just in Canada, but in other countries that are especially highly vaccinated. And many of these relate to cardiovascular disease increases. And what we're seeing, increased rates of cancer that, again, we cannot account for, but is coincident with the introduction of these vaccines, not in 2020, but in 2021.
How did you feel as a scientist to watch our government and the media tell us our own immune systems were worthless compared to the vaccine?
Yes. Well, people like Bonnie Henry, our public health doctor in British Columbia, proclaimed just that she doesn't know about natural immunity. Well, why are you in this position if you are not aware of our body's best defenses, which is, this immune system is so amazing in what it has done through evolution in animals over millions of years. And even a starfish has an immune system. And that's about 1 billion years separated in evolution from people. We've been there before. We've been dealing with viruses and bacteria with knowledge for over 100 years of vaccines being developed, and how the natural immune system operates. And we have huge institutions around the world, and with people like me that are studying this on a daily basis. Why someone would make that kind of statement is ridiculous. Even SARS-CoV-1, we know that people who were infected 22 years ago have antibodies today against SARS-CoV-1. I know that for a fact because I tested it myself. And in fact, this is what's very interesting. We find that when we tested people and we identified the parts of the virus that were the most immunogenic, that is the most likely to develop an immune response, and we took those same regions in the viral proteins of SARS-CoV-1, we found that those people that recovered from SARS-CoV-2 had stronger reactions against SARS-CoV-1 than SARS-CoV-2, which basically instructs us that these people were exposed to a SARS-CoV-1-like virus earlier in their lives, maybe even recently it's been in the environment. And then what's happened is those immune cells, T cells and B cells, those populations that recognize that were expanded. So we had a lot of those cells. So when this new virus came along, which isn't really that new in terms of its actual structure, those antibodies that are produced by those B cells and those T cells were able to recognize the new virus. And then they become amplified further. So that's why we have, stronger responses to the original SARS-CoV-1 than SARS-CoV-2.
Wow.
And that explains why many of us, right when the first time we were ever exposed to SARS-CoV-2, didn't get sick.
Okay, so can you talk about the mutation because the mutating, as they went through the variations of the virus, and unvaccinated people were villainized and blamed for these. They were going to cause these mutations. I don't know. I'm not a scientist, and I always thought that like something mutates to get around or beat something. So I was always of the mind of like, if I'm unvaccinated, it can get me. It doesn't have to mutate to get me, but maybe if I'm vaccinated, it's got to mutate to get me. So, like, can you just talk about that or put that some light on that there?
Well, as it turns out, if you're vaccinated or not, if you get sick, you have very similar viral loads, and the duration of the illness will be about the same. So the pressure that's there on the virus is actually more the ability to spread and not make people sick. So that way there's a better chance that it can go from one person to another. If you're sick, you stay home. You're not going to be out there spreading it. But if you're unaware that you've been infected, then there is a better chance that you will do this. And so this is how the mutations are such that they actually allow it to become more benign and more infectious. But these mutations, what most people also don't understand is that when you are infected and you have all these mutations occurring, most of these mutations actually compromise on the ability of the virus to actually be infectious and replicate. But a very small portion can, and those are the ones that are going to be problematic for us. So it is a natural progression, basically, that occurs with any kind of, pandemic. You will have a portion of the population that succumbs to it, and then you're left with a population that's actually resistant because of either their genetics or their previous experience in their lives of being infected and recovering. And that's just the way it's always worked. For example, you'd have the deadly in 1348, you'd had the Black Death, wiped out about a third of the population. Maybe some people argue even a half the population of Europe. But people were resistant as well. And so there is this natural diversity in our population genetically. And viruses have to actually evolve to very specifically bind to proteins that are on the surface of the host cells. If they can't attach, they can't get in. And so you might be astounded. You have about 50 trillion cells in your body. And you have about 40 to 50 trillion bacteria cells in your body, but they're much, much smaller than your normal cells that have your DNA in them. But you also have about 350 trillion viruses in and on your body. And most of those are actually pretty benign. Some of them are actually quite helpful. They kill bacteria, for example, that might be pathogenic. So, as a colony of all these different cell types and viruses, we're healthy. And in fact, we need some of those bacteria to produce vitamin K and to fight pathogenic bacteria. Because when you take an antibiotic, you wipe out a lot of the bacteria. But if you have an antibiotic resistant bacteria that's pathogenic, you actually increase your chances of getting sick again. So our immune system learns to recognize what's problematic and what it is, okay, it's benign. It's actually maybe necessary. So when you look at the total number of viruses that are out there that can infect, as it turns out, mammals alone, we can count already probably in the order of over 300,000 different viruses that are actually in the environment that can infect mammals. And some of those infected mammals, if you have mutations, might allow it to jump from the animal to people. So this is actually extremely common in terms of the potential, but the actual rate at which most of those viruses are going to cause us a problem, it's so rare. The number of viruses that are actually pathogenic to people is probably less than 100. So this is the thing. It's not that easy for a virus to evolve to specifically get inside a cell and then hijack the cellular machinery, the proteins that would normally be used for the cell's purposes, to make it into virus factories to produce more virus. Everything has to be very precise. And so those criteria are so strict by which you can have that happen. It's very rare.
But what we are seeing is in the vaccine, if you have multiple boosters, you've essentially set up a spike protein factory in your cells.
That's correct. And so one of the concerns that I have is that, well, okay, we've dealt with SARS-CoV-2, and there is some efficacy for a short period of time with these vaccines. There's the risk of injury as well. But what happens is, right now the industry is looking to make more of these RNA vaccines against other infectious diseases. And not just these RNA vaccines for the human population, but we want to do it for the animal population too, for cows, and, for example, to protect against H5N1, which is the latest concern. Even though I think the number of people in North America that have actually been infected with that influenza virus is somewhere around 14, one in British Columbia. But the concern is that, well, we've got our eyes on this with so few people actually been infected, and we don't even know how that person in B.C. got infected. So
that is in Bill 293, looking at taking over control of livestock and farming and researching alternative proteins. That was correct. Like, wow, cart before the horse there. Like
well, it's a one world view that we're one system. So it includes not just humans, but animals, silviculture, like the agriculture, the plants, as well as the animals. And so it gives wide sweeping powers to the government to decide how they want to, discourage, will be the poultry industry and maybe, for meat consumption. I'm a vegetarian. I've been a vegetarian for 40 years. But at the same time, I recognize that what they're trying to do, it it's just giving them extra powers that they can use for other aims or other means.
Because I
And this,
would know whenever there's a health outbreak, a lot of times it's lettuce.
yes, yeah,
Salmonella, Listerin, or whatever. There's a lot of bacteria breakouts on those, right?
that's right. Well, we have factory farms,
Yeah.
and what we're essentially doing is monoculture, and that increases the chances that if you have a an infectious agent, that it might actually wipe out the entire crop. This is what happened with potato blight in Ireland.
Mm.
They all planted potatoes, and then it turns out that they have a bacteria, I think it was, or maybe it's a fungi, and what happens is it just wiped out all the potatoes, caused massive starvation because you have this monoculture type, you know, instead of having a diversity of different types of crops that are growing in a more of an ecosystem that can include animals along, for fertilization purposes, um, to make the soils more fertile. And you have this queer situation where you put all your cows into certain fields and they have all the manure and everything. And meanwhile, you have other fields where you want to cultivate crops, but it's not getting the manure from the cows because they don't want the cows to walk all over where the crops are. So what we have to do is we have to develop systems that are actually enrich the soil naturally as whole ecosystems. But that's another story.
I know. Okay. My next question, long COVID. Can you explain COVID or give me your thoughts on long COVID? My big question is, do you know any person who is unvaccinated who is suffering from long COVID?
Well, we do know in the case of long COVID, we do know that people early in the pandemic, before the vaccines were introduced, did get long COVID. So it is a consequence of viral infection. And what we observed early on is that the people that got long COVID generally had comorbidities to begin with, and these were exacerbated with the virus infection. So, so, so we do know that it is a consequence in part from the virus infection, at least in 2020. Once we get to 2021 and beyond, where we introduce these vaccines, many of the symptoms of long COVID are very similar to vaccine injury as well. And many of those people that developed long COVID, what we're seeing is that we can sometimes treat those conditions with Paxlovid, which is a very specific inhibitor of the virus's replication. And there's also been success with ivermectin as well. So what that seems to be indicating is that these people with long COVID may have persistent infections with SARS-CoV-2 at a low level. And they're not getting rid of it. Their immune systems do not appear to be able to completely eradicate the virus.
Or because I'm in some Facebook groups of people that have long COVID
Yeah.
and they are obsessed with COVID and a lot of them are obsessed with boosters.
Yeah.
So could some of them find themselves just in a bad treadmill where yes,
Well,
they've
this is
developed
exactly
something.
it. We see that the more boosted you are, the more you depress your immune system against that virus. So you're tolerizing your body to the virus infection. So it's not going to be as efficient at removing the virus. And so you have this persistent low level infection, which then further exacerbates the chronic condition that that person is in. So this may not be helping them. It could well be that using certain medications like the Paxlovid, which, there is rebound that occurs maybe in 5% of Paxlovid cases, but it is very specific for the virus, and it does actually help. But the other thing you can do is boost your immune system, and you can do that naturally in part, your diet, exercise, sleep, having a sense of well-being, that you're not stressed, all of that can facilitate your immune system to work better to get rid of the virus. And if it's a vaccine injury where you've got the spike protein is being still produced and you're getting attack on your body cells, again, your immune system is your best bet to actually removing those cells. So, and for most people, this will happen on their own in a relatively short period of time. But for these people with long COVID, there's some compromise with their immune system that it's not working as efficiently as it should be to get rid of the virus and to get rid of cells from the vaccine that are still producing the spike protein. The problem is the SARS-CoV-2 virus not only binds to the ACE2 protein, but it actually binds also to other proteins, including neuropilin. And because the lipid nanoparticles can pass through the blood-brain barrier, they're able to infect nerves. And so you may actually have a variety of symptoms that reflect more autoimmune disease attacking, the nervous system that can give rise to some of these. I mean, there depends on the organ that is most affected by where those lipid nanoparticles went and that person's unique situation of whether there's already underlying autoimmune disease going on. They may not, it may have been very mild, but it's just exacerbated by creating a perfect storm here. So we know, for example, there was one study that was done with a Parkinson's patient. It was reported by Dr. Mortz out of Germany. He was a pathologist. And he looked at a person who died of Parkinson's disease. And interestingly, apart from seeing and this was months after they were vaccinated with the Pfizer vaccine, he could see in the autopsy, we was doing what we call immunohistochemistry, basically of the brain regions. He could see spike protein present, and he could also see that the immune cells in there, we have immune cells called microglia, that they're attacking that region where there's damage. But interestingly, he also looked at the heart of that individual. And that person also was producing spike protein in their heart. And they had infiltration of the neutrophils, macrophages, at least monocytes of some kind, their immune cells, in that region. And so this might be part of the underlying reason why people develop myocarditis. And in the case of myocarditis, what you have is the chances of arrhythmia in the heart. And so the heart isn't beating in unison to allow the blood to flow through the chambers, but it's actually pumping against itself. And of course, that happens, people faint. And so the long-term consequences of myocarditis, and this could be asymptomatic myocarditis, okay? So you don't necessarily have it where you fainted. And that tends to happen in two types of periods. One is when you're very physically active. So this is why, males that are athletes probably at greatest risk. But also when you're sleeping in the middle of the night, you get surges of certain hormones that actually can at the same time trigger these kind of responses. So the heart is actually with the myocytes, these are the muscle cells that are contractile. They allow the beating of the heart. Well, what happens is they're killed off by the infiltration of the immune cells, and you're left with scar tissue instead of myocytes. The neurons in your brain and the cardiomyocytes in your heart is one of the few cell types that are not replaced by the same type of cells. In your neurons, the average neuron is connected to about 10,000 or more other neurons through what we call dendritic connections. This is where all your memory and learning is stored. So you don't want to break those connections, because if you did, and you have to do that if you're going to have these cells divide into daughter cells that can replace the dead cell, you can't do that with neurons, and you can't do it with muscle cells of the heart. They're bundled. And if you were to have these cells break their connections to allow them to divide and repopulate, you just can't do it. So it's scar tissue. Surrounding muscle cells need to get bigger to carry the load to pump that blood through the heart. And with a bigger heart, initially it provides you with, functionality, but in the long term, it increases your chances of arteriosclerosis and then further clogging of your veins and arteries, making your heart have to work harder and bigger, and then you just set yourself up for cardiovascular disease. So in Canada, the number one cause of death is cancer, but the second most cause of death is cardiovascular disease. In the United States, it's the opposite. They're more likely to have cardiovascular disease and then cancer. But these are the number two killers, basically first and second, for what most people die from. So you're setting up people for cardiovascular disease, neurological disorders. You're interfering with your immune cell function, so you get autoimmune diseases, and you also have less efficiency of your immune system to recognize cancers, as it turns out. So this might account for why some people observe what they consider turbo cancers. But for sure, we have increased rates of cancer since the introduction of the vaccines by about 10 to 15% for at least 2 years. We don't know how long that increased trend is going to continue. The last time I looked at it, we had data, it was still increasing in the population. Yeah, this is the big concern, that we're not seeing things getting better. We're seeing things getting worse. So I think, in the United States, with the election of Donald Trump, he's made a commitment to try to improve the health of the American population, which, is one of the most unhealthiest in the world, high degrees of chronic disease. I mean, even our children in Canada, we're looking at about 40% of our children have chronic disease. We didn't have this, a couple of generations ago. And of course, it's even higher in the adult population. So this is where, with Robert Kennedy Jr., for example, a very strong advocate of children's health. And I think this is where we're going to be able to see maybe some change, so that we're better resistant in the future to this. But meanwhile, we have to encourage people to, take a hold of their own health and do what they can themselves to protect themselves. And it isn't a mask. It isn't staying home. It's going out there, getting exposed to these viruses, educating your immune system, eating healthy foods, interacting with people you like. That social interaction is, again, one of the secret ingredients for why Japanese people live for such a long time. They care about each other and they're very social. But we have people in our societies that live in apartment buildings right next to each other, and they don't know who their neighbors are. I mean, this is the way our community has gone, very different.
The origin of it, you don't have to go deep on this, but are you just suggesting that both origins of the virus could be true, that it could have started in the lab and then somebody who works at the lab goes to the market, and the market's a big public space, and then it's kind of...
Yeah, the origin of the SARS-CoV-2 virus is
Because
complex.
you were saying that COVID-2 seems to have been synthesized, or is there anything? I don't want to
Yeah,
put words in your mouth, but
so like I say, the origin of the SARS-CoV-2 virus is complex. I think everyone is in agreement that it's most likely to evolve from a bat coronavirus because of this 96.8% genetic identity, almost identical. These very subtle differences make the difference between whether it'll infect people or not. SARS-CoV-2, that virus that, we considered, the Wuhan strain, it has features that are like SARS-CoV-1 that are not found in that bat virus. We know that Ralph Baric in the United States was proposing experiments to actually do genetic engineering of the bat viruses to make them so that they could infect people. And again, the rationale behind this is that they want to prepare themselves for a future pandemic by, having these viruses so you can start to develop vaccines, for example. So the Ralph Baric's graduate student later became the director of the Wuhan facility. And a lot of that came about because of the SARS-CoV-1 came from China. They put a lot of effort into developing the capability to study bat coronaviruses and SARS-CoV-1. And so she brought her knowledge from Ralph Baric's lab, and they clearly were doing genetic engineering at that facility. Now, as it turns out, they built what we call a biosafety level 4 laboratory, the only one in China, aided with the help of French scientists originally. And there is evidence that, in fact, they were working with SARS-CoV viruses, SARS-CoV-1, and other coronaviruses, like from the bat, for at least 10 years before SARS-CoV-2's pandemic. And they have videos of where they're showing in the facility them working with bats, even though later they denied that they were working with bats. And there's publications that show that they were working with bats and doing genetic engineering. So there are features, like I say, in SARS-CoV-1 that you can find in SARS-CoV-2 that are not in the bat. So I think they're guided by experience with the SARS-CoV-1 to guide them in how they do their genetic engineering on that bat. And so I think it's pretty clear to me that it's most likely that that particular virus was from a lab leak. There's actually two strains of the Wuhan virus, an A and B strain, that appeared to have arose from separate events. So there may well have been more than one release of that virus. And there have been reports of over 200, even in the United States, you have lab leaks happening all the time, usually about, could be up to 200 or more reported leaks of people working with viruses in these kind of level 3 and level 4 facilities. In Canada, we only have one level 4 facility, and that's in Winnipeg. So, but they had level 3 facilities in many cities that were in China, and there was even a level 2 facility that was only a few hundred feet away from the Wuhan market. And it's not clear that they would have necessarily done work on these coronaviruses only in level 3 and level 4 labs. I think it's pretty common. I mean, this is the irony, with the masks, right? If you want to work with SARS-CoV-2 in research, you have to be in a level 3 or a level 4 lab. And at the University of British Columbia, we have a level 3 lab. We're not allowed to work with this virus for doing research unless we have a level 3 or higher. Now, in these labs, you have to have like a bodysuit. You have to have separate oxygen. You have to be working in laminar biosafety cabinets and negative pressure rooms with a mask and face visors. Okay. And yet our government was telling us that, yeah, you can go ahead with a cloth mask. Theresa Tam was saying that, you cut a piece of cloth and you attach, basically string to the cloth and you wear it, and
First,
that's a
first,
mask.
first
And
she
to
came out
protect
and
you against
said,
this virus.
first she came out and said masks don't work,
That's right.
and then she did a 180 and said,
Oh yeah, and likewise with Bonnie Henry, said they didn't work, and then yeah, now you have to, a day later, after she's saying they're not working, she says, "Oh, we have to introduce, the Fauci said the science changed, but basically the next day she said this, and then we have this situation now in BC that the uh, the healthcare providers have to wear a mask in the hospitals, but the patients don't, and the visitors don't. I mean, I don't understand the logic of this. Of course, they don't work anyways, but nonetheless, these are largely um, made on the fly, and it's a way of government seemingly taking decisive action to protect its population. And even though at the end of the day, this is causing more grief than actual providing any remedy to the situation.
Canadian government, always sure, often wrong.
Well, I would give credit to a lot of the people that are working in Health Canada and the Food and Drug Agency. I think that it's driven top down politically. I think they are pressured to go a certain way, um, and unfortunately, they have to succumb to that pressure. But I think most of this has been politically driven, unfortunately.
It's scary to see, all of what we know through
Mhm.
research. And, I know the research seems to be slow getting out in Canada publicly. I know other countries, America,
Oh
cases
yeah.
are being brought, but it feels to me like our politicians and our health, I guess Health Canada, have doubled down. They're not acknowledging anything. In fact, they're just it's booster season. Like no one's even stopped to say. I mean, we have a massive problem with our government double downing. They can never
Well, it's so
anything.
bad. For example, Teresa Tam was a co-author on a paper that was published in a peer-reviewed journal, Canadian Medical Association journal, and they did what's called modeling. It's counterfactual modeling. It's where you're taking pre-existing data and you're working backwards, and you're making an assumption that you didn't do something, what would have happened. So in this case, this article was published on the idea that had the government not done the lockdowns and the masking and the vaccination, how many people would have died in Canada? And so in this modeling, basically, they say that had they not done, and it's based on certain assumptions, which are actually you can challenge very easily, but they're basically saying that had they not done this, that up to 800,000 people would have died in Canada. Now, put that into perspective. That's more deaths than for Canadians than the 1918 influenza pandemic, World War One, and World War II, adjusted for a smaller population, combined. The number of people that died in Canada, we're looking at somewhere, I think it's around 7,000. And of those 7,000, they died with COVID-19. So it's probably even half of that. And of course, the United States, it's crazy, their estimations. And this is very interesting. If you take the number of deaths per capita in Canada and you compare it to United States, we're about 7 times less deaths per capita. But if you take the number of deaths in the United States and you compare it to the number of deaths in China, where this virus started, you have 870 times higher deaths per capita in the US than in China. Now, you can't say, well, it's because the Chinese didn't report, all the deaths. I'm sure that there was a bit of that suppression by the Chinese communist government, but in most cases, even other countries in Asia, you're still seeing, could be 100 or 50 times less deaths per capita in those other Asian countries, so South Korea, Japan, than you're seeing in the United States. Same virus. And so it's a reflection, I think, in part, you are more likely to get, you not even have to pay your medical bills, which are very expensive in the United States if it's a COVID case that you had a death. But the other is that the health of the population is so poor in the US relative to these other countries that when a pandemic comes along, they're just way more vulnerable. And that's why it's so important to change the healthcare system to promote wellness versus a counter response after the fact when the basically the cow was left the pen, the bulls left the pen. We need to actually build a better fence so this doesn't happen, and that will happen when we have a healthier population.
Unfortunately, it's going the other way. Asia is tending to replicate us with
Yes.
diet, and because they want to become, yeah, I mean, it's just the way that it looks, that how a society, it looks to be the way a society develops, but it's not, it's garbage. But and Teresa's TAM modeling, wow, that's scary. That reminds me of like economics.
Yeah.
Economics, there's a reason economics is a soft science, because its modeling is highly sus. And if I hope that health science does not go that direction.
Well, I'll tell you the problem with the modeling, and probably one of the worst abusers of this is David Fissman in the University of Toronto. And he's modeled that it was the unvaccinated that was responsible for the spread of COVID-19 in the vaccinated. I mean, I've written articles on this, and in our book, Down the COVID-19 Rabbit Hole, we actually explain this. And in particular, we have the second book, which is more on the politics of COVID-19. That one's called COVID-19 Pandemonium, a pandemic of ignorance, fear, and greed, the capture of our institutions. That's where we really go into the modeling problems. But they're based on certain premises. Now, many of the people that are doing these modeling, they're very good in mathematics, but they have very little knowledge, it seems, about immunology and virology. So when they put their assumptions into their models, the assumptions are wrong. And so, garbage in, garbage out, and that's what happened. But unfortunately, these these models, and Neil Ferguson in the United Kingdom was one of the first to really go at this in a big way, predicting tens of millions of deaths, early on in the pandemic, that just frightened the population and especially the politicians to take these measures that at the end of the day were very harmful to the population. But based on those modeling, that's how we got interim order approval of these vaccines, and likewise the emergency use authorization in the States.
What do you think, looking at the lay of the land now, and you've written two books, so clearly you're invested in this. What do you think needs to happen in Canada?
Well, I think firstly there has to be an acknowledgement that we did not handle this very well, and we have to reevaluate the science. And we kind of tried to do that in the two books I was involved in writing with my colleagues, primarily at the Canadian Citizens Care Alliance. We had about 24 authors on these books, but we went really delved into the scientific literature. And, we had about 1,100 citations of the scientific literature in the first book, Down the COVID-19 Rabbit Hole, and about 800 in the second book, which, again, COVID-19 Pandemonium. And so we're trying to lay the basis of, what exactly happened. There has been inquiries. There was the National Citizens Inquiry in Canada. The government has undergone an inquiry that it just published recently. It's called The Time to Act. And as we go through that document, we recognize that they did not really deal with the science of what was going on. And it pretty much is party lines, right? It's very political. So we're actually going to come up with a response to that. But our books actually have this information. And so we need to get more public officials and more politicians elected, like happened in the United States, that are willing to take the bull by the horns, recognize the problems, and start to take measures that are actually going to be effective solutions to future pandemics without eradicating the human rights that we also have and hold so dearly, which unfortunately was really sacrificed during the COVID-19 pandemic. We have two victims here. We have the people that didn't get vaccinated, and they lost their jobs. They were vilified, and a lot of economic, hardship for these people. And then we have the vaccinated individuals that, did what they thought was being good citizens and wanted to protect themselves and their loved ones. And they had trust in their government. And a few of them, most people will be fine, but maybe one in 400, 1 in 500 had permanent damage from taking that course to protect themselves against this virus that, at the end of the day, wasn't that much worse than a bad season with the flu. The other is that there were many natural remedies that were available early on, like ivermectin, hydroxychloroquine, vitamin D. Vitamin D is the best thing you can do. You don't need a prescription. If you're living in Canada, the odds are that you are vitamin D deficient in the winter months. We have less sun. When you have sun, you only need 10 minutes of exposure to sun on your skin to give you all the vitamin D that you need. So in the spring and certainly the summer and early fall, it's fine. But during the winter time, you should supplement yourself with vitamin D. And the 400 unit dose that's recommended is based primarily on what you need for your bones. But for your immune system boosting, you probably need about 4 times that. So I would recommend having vitamin D, and you don't build your vitamin D immediately. It takes a week or two to build up the levels that will be sufficiently protective. But having a supplementation with your meal is once a day is probably not a bad idea, especially if you're a little bit older. So I would recommend that.
Any thoughts on the vaccine support or the vaccine injury support program? Have you heard anything of that?
Yes, so we have the KFIS system in Canada that's supposed to be keeping track of injuries due to the not just COVID vaccines, but other vaccines. But primarily, when you look at how that system worked, you would think that there was hardly any vaccine injuries in Canada. And there's a problem here because the phase 3 clinical trials with Pfizer showed that up to 5% of the population or the participants that were vaccinated actually had severe adverse events. So you would expect that somewhat in the general population. Now, there is some differences between the vaccine that was used in the clinical study. The manufacturing process was very different from the one that was used for the mass production. The mass production one has higher amounts of contaminants, bigger opportunities for toxins to be included, endotoxin, for example, has DNA plasmid, which, the DNA plasmid is your lipid nanoparticles are supposed to be primarily RNA, but some of the DNA plasmid used to make the RNA in the bacteria that in the processing initially contaminates, could be up to 1 to 2% of the total amount of genetic material that's actually in the lipid nanoparticle. And the problem with the plasmid is that it means you can keep producing more RNA continually. So this is maybe why we can get in some cases persistent production of spike protein even months later. So there is that issue. So if you have a vaccine injury with the CAFIS system, what you're supposed to do is the doctor, could be a nurse, has to fill out a report. Takes 20 minutes to fill out. And then it goes to a local center for review. And there is this special criteria that has to be applied for the timing of the report of the injury since you were vaccinated, and all these criteria that are actually set up, criteria with guidance from the vaccine producers themselves. So what happens is that's vetted locally, and then it goes nationally to the Health Canada, where basically the CAFIS system is operated in part from, and the Food and Drug Safety Administration of Canada. And what happens is it's further vetted, and if they don't think it meets these criteria, then basically it doesn't get included. Okay, so it's very, very strict criteria. So if you have other symptoms, for example, that haven't really been previously associated with that vaccine, it might be excluded. Now, we have doctors in Ontario and also in British Columbia that face disciplinary action. Patrick Phillips would be the one that did this in Ontario, for example, and his college reprimanded him for filing these reports, as is what's going on with Charles Hoff and other doctors. So there's a strong disincentive to report these injuries, that these doctors could lose their licenses. And they also have to look at the sacrifice of income. We figure that the average doctor in Canada that prescribed these vaccines, they actually made a lot of money. In the United States, you can calculate, could be as much as $250,000 extra in their salaries by offering these vaccines. And in fact, doctors were paid way more for with the Blue Cross and that for prescribing these vaccines than other people. Now, of course, we have a situation where you can get these vaccines in a pharmacy, without a prescription. And so you got these, how's this pharmacist going to know what the health history is of this patient to make the decision that, yeah, the benefit of the vaccine is greater than the potential harms? That person may have pre-existing myocarditis. That person could have a number of other ailments that could be exacerbated by the vaccine shot. And how many vaccine shots have they had? So instead of the doctors doing that consulting and explaining to the patient, informed consent means that you recognize the pros and the cons, the risks and the benefits. So, we've actually even taken that away from the doctors. So there was a lot of carrot for the doctors, and there's a big stick that's there for the doctors. So they're going to be very reluctant to say anything against these vaccines. And even the scientists like myself that are in universities, most of us are funded in our research from the government or from pharmaceutical companies, sometimes from charities. But our livelihood depends on us getting grant funding to do our research and publishing. And if we can't get funding, we can't publish. And if we don't publish, we don't get promoted. We don't even necessarily retain our jobs. So there's a huge, barriers for other people that are in my position. Well, my position is a little better because I'm close to retirement and I'm tenured, and I have a company that pays for my research that I do. So I'm kind of a little bit insulated from this. But most of my colleagues, they're very vulnerable and exposed. And so they're going to be very careful what they say, and they'll have to couch. We had scientific papers that were in the last few years that come out. The beginning of the paper says how great, how deadly the disease is, how many people died. And then, vaccines have been the best, strategy to deal with it. Then they do their study showing all the reasons why these vaccines aren't that good. And then the final paragraph says, it's still the best thing that we have to fight this disease.
Yeah.
I call these sandwich papers. The meat is in between, and that's where it's telling you, hey, this is not so good. But the top layer and the bottom layer, beginning and the end, are going with the narrative.
Wow. I think I recall reading some articles that were based off studies like that. Yeah.
Mhm.
And it was yeah, it was kind of a mind burn, like what is going on here? And so I guess you explained why you haven't been canceled. I guess you're insulated there. So that's
Yeah, no, I've been very fortunate. I've had many of my colleagues, though, including in the Canadian Citizens Care Alliance Scientific and Medical Advisory Team, that they were either unpaid suspension for years, and some of them were fired, like Patrick Provost in Quebec. He was fired for speaking against the vaccination of children from his university.
I reached out to the Ontario College of Physicians because I said, doing a documentary on a vaccine injured gentleman, and many of his doctors won't talk to me because they think you're going to take his license away. This has got to be nonsense, right? Will you talk to me and explain your side of the story? They said,
Yeah,
sorry, we can't talk to you.
no, I
I can
think
only hope that the vice is tightening around them, that they feel, and I don't know, because I'm very disheartened as a citizen here, um, that this is our healthcare system and the power that certain departments wield here against our lives.
well, again, it's politically driven. I think most of the people that are in these professions, or in these departments are actually quite competent, but their hands are tied. We saw the same thing with the media. I've interviewed many reporters that are pretty awake, but they just can't get past their editors because the editors themselves are basically being controlled at the highest levels, in part through government. I mean, you've got government spending like $1.2 billion a year in supporting the CBC, for example. And the advertising revenue, I think, for the CBC is somewhere around 0.5, 0.6, sorry, a billion. So most of the money to keep that institution, CBC, alive is coming from government. You are just not going to criticize the hand that feeds you.
So, are the vaccines safe and effective?
No, these vaccines are really not safe, and they're not effective, because when you have a vaccine, you're giving it to healthy people. You're protecting them against future infection, such that they will not get that sick, certainly not sick enough that they're going to require hospitalization or could die. The reality is that these vaccines are fleeting in the immune protection they provide. They also, at the same time, have this risk because you're attacking your own cells of causing damage and exacerbating pre-existing conditions. So most people are fine, but a small percentage, maybe 1 in 500 people, 1 in 400, something in that range, seem to be very adversely affected. There's also something else that's worth considering. This is in the estimations of the efficacy of these vaccines. The idea is that you're comparing unvaccinated people to vaccinated people. But what constitutes being unvaccinated is that even if you've been vaccinated, and in the first shot in B.C., the first 3 weeks after you've been vaccinated, and remember, most of these vaccinations occur when there is a pandemic, when you're in a wave of infection. Most of those, like the first 3 weeks, if you get COVID-19, you are considered unvaccinated, and you are lumped your data with the unvaccinated population. So you're skewing the data. Now, at first blush, you don't really recognize there's a problem here. However, Alberta Health published data, and they had it up for about 5 months, where they tracked what was the rate of getting COVID-19 if you've been vaccinated from day one. And what they showed was in that data that in the first 7, 8 days, the risk of getting COVID increased daily as after you got vaccinated. And then it stabilized. And then after about 10 days, it starts to drop down, presumably because you evoked an immune response that's going to give you some protection. So you actually, in that first 3-week window, are increasing your chances of getting COVID-19 when you get vaccinated and you go out where the virus already is. And people wonder, well, how does that work? And the reason is quite simple. When you are infected, the kind of antibodies that you make are IgA and IgM antibodies. We call these mucosal antibodies. They're produced at the sites of the nasal pharynx, the mouth and the nose and upper lungs. And they're very strong and protective against viruses and bacteria. And they clump very effectively these so that you can cough them out, or you can lose it with postnasal drip. So they're very effective antibodies, and they're very different from the antibodies that you produce when you're injecting the immunogen into your muscles, which then again spreads with these lipid nanoparticles. There you're producing IgG class antibodies. IgG class antibodies are very good antibodies in the sense that they last about 21 days compared to about 4 to 5 days with IgM or IgA antibodies, but they're not as effective at clumping up these viruses and bacteria and getting rid of them. And the concentrations of IgG antibodies that you have in your lungs and upper lungs and airways is much, much lower than the IgA and the IgM antibodies. Now, imagine a scenario where you have the virus in the environment, you're in the middle of a pandemic wave, and you go and get vaccinated. Well, any immune capacity that you have that's going to recognize that virus, especially for the first time, is going to be working with whatever you have got that may have been recognized previous infections with coronaviruses, for example. And your, what we call your innate immune system. This is the very mobile part of your immune system that's non-specific, but it's very important. Macrophages, neutrophils, they clean things up. Well, if you've got producing on the cells of your body quadrillions of copies of these spike proteins inside your body, then your immune system has been remobilized to fight where it thinks the infection is the worst. So it's like a castle with guards where you've got lots of guards throughout the castle ready to fight, the intruders. But at the gate, at the front of the castle, it's open and there's no guards. So the virus gets in, replicates, and at that site also spreads. So you are more likely to actually get infected. And we see that in actual data. And this is one of the reasons why that data with those vaccinated people in the first 2 to 3 weeks are lumped in with the unvaccinated and then used to justify that, oh, if you're vaccinated, you have less of a chance of getting COVID-19. Of course, as you have repeated booster shots, you're actually getting negative efficacy, and the period of protection gets shorter and shorter. And so this is where we're at today.
Okay, last question then, because I don't want to take up any more of your time, though. Going forward with something like this, where do we go from here?
Well, the vaccines themselves are actually still a good strategy to try to prevent illness. I think that we overdo it. We're as much as 70 shots for people before they're even like 10 years old. It's crazy because of the risks versus we don't know what happens when you're doing, especially at that stage in life when your immune system is getting educated and you're trying to distinguish self from non-self. And when you're not exposed to these things, it's like peanut allergies, for example. People get anaphylactic shock if they don't have peanuts very early on in their life, many people. So you need to get exposed to these things, but you the concern I especially have is that we're switching from these traditional vaccines, which are much safer and we have a much better track record, to now adopting this new technology with these RNA vaccines. And we recognize already now that the adenovirus vaccines are not very effective. They have their own problems, and they can only be used once or twice, because then what happens is they're using a virus to deliver the DNA. And with that delivery with the virus, you actually develop an immune response to the virus itself, the adenovirus. So really, it's RNA technology with lipid nanoparticles that we're pushing towards. And there is a lot of vaccines that are in development right now that are RNA vaccines. And at the University of British Columbia, for example, it's going to become a major hub for the development of these vaccines. So the issue is that, well, the next vaccine may not be producing spike protein, but it's going to produce another protein from this virus or bacteria that you're trying to develop an immune response to that ideally should be protective. But you're still attacking the very same cells or other cells in your body. And the consequence of putting in that protein and having that immune attack against your own cells means that your first shot with another RNA vaccine for a different virus, if you've already had 3 or 4 shots with the COVID-19 vaccines, this is like your 5th shot. Mm-hmm. So it's like Russian roulette. So at some point, the bullet in the barrel is going to line up, and you're going to have a bad effect. And so this is why I think this technology is actually doomed. I think we will recognize in the long run that this causes more harm than good. Hopefully we can go back to some of the more traditional vaccines that are effective. They last a long time. They're safe. And I think what's happening is we recognize now, though, we need more testing data. We can't just go based on a limited amount of data, give it to the wide population. So I'm hoping that where we go from here is that first people become aware of these problems, that politicians, recognize this as well. We're already seeing this in our population. I mean, the fact of the matter is the adoption of these booster shots has just plummeted, as has the stock price of Moderna, and Pfizer has gone down to its pre-COVID stock prices now, where they were making as much money, from COVID-19 vaccines as all the other revenue we're developing from drugs that they offer, right? But this is all kind of dropped. People are not getting these vaccines. They very rarely know someone who's died from COVID-19, but they often know someone who's had a vaccine injury. And so the public knows it. In many other countries, in Europe, for example, they don't recommend these vaccines anymore for children. They don't recommend for even adults. And in some places, they won't even pay for it as they had done in the past. And this was already happening well over a year and a half ago in Europe. In North America, we just do not seem to be getting the message. And so in the United States, the population has voted for a new government that's going to pay attention to these and not feed them misinformation, frankly, and fight against the misinformation about what has been called misinformation. For example, what I'm telling you now, I'm sure some people will say, well, this is misinformation. No, you can look at the scientific literature. Everything I've said to you, I can demonstrate from either personal experience where I have the data that I can show you, or it's from published peer-reviewed scientific journals. And this is being recognized around the world with many more scientists that are coming around, and health professionals. I've never encountered somebody who was dead set against these vaccines and was concerned that now is feeling better about them. I'm seeing the opposite. I'm seeing people, that were gung-ho about them originally, but now they don't advocate it.
Great. Well, this is fantastic. Thank you so much for taking the time.
Well, hopefully there's some material there that you might be able to use.
I might maybe get 30 seconds out of it.
Yeah, that's what I figured. Maybe about a minute if I'm lucky. But that's
Well, this
okay.
is really great.
That's the nature. And that's what you should be doing. You should be talking to different people with totally different opinions. And somehow, when you see what they have to say, you kind of start to get a sense of, the truth is somewhere presumably in between. I think you'll find it's a lot on my side because I've done so much work. I mean, I have nothing to profit from this. Even the books that we produced, all the revenue goes to the Canadian Citizens Care Alliance. I don't have a salary from that organization. My saying this stuff, it's only going to give me, some grief from my colleagues. So there's nothing for me to gain from it. But what I am very concerned about, and this is what scientists should be doing, right? We're looking for the truth. We want to know what's going on. We don't want to be going down and dogma that actually is false. We work hard to fight against dogma and seek truth. So I'm just doing what it is that I'm supposed to be doing as a scientist.
Yeah, absolutely. It's interesting that you say I should get a varied voices of opinion, which is yes, as a filmmaker, that's my number one thing. But this is probably the one project where it's the most difficult. Yeah. So
Interview Timothy Caulfield. And so Timothy Caulfield is a very, very active individual. He's a professor. He has no actual training in health sciences at all, or even science from what I can tell. And he's one of the main spokespeople. He's had like huge grants to go out there and challenge misinformation. and he will not actually debate anybody who has a different opinion from him, and his opinion is completely informed by Health Canada and government. But he has grant funding to
Yeah.
basically go out there and call people like myself purveyors of misinformation. And it's kind of interesting. The Vancouver Sun wrote an article, I guess about 2 years ago, where it knew that I was concerned about these vaccines, and they kind of, I guess the reporter decided to try to make it look more like I was like a boxer or something looking for a fight. And they got Timothy Caulfield to comment on whether he'd be willing to talk to people and debate these things that he's called misinformation. And he said that he won't do that because he doesn't want to give a platform to people that actually are spreading misinformation. So, but he has written so much garbage, it's incredible, and he's being paid by the government to do this. But I would
Okay.
actually interview him if he's interested, because he's appeared in a lot of programs and podcasts, and he has TV, and he's seen as sort of like a poster boy for let's go for these vaccines.
That's who I need then. That's who I need.
Indeed. Well, thanks again, Dean.
All right.
Good
Thank
to
you.
talk to you. You
Pleasure to
take
talk to
care.
you. I really appreciate it. Have a great weekend.
You too. Bye bye.
Bye bye.